Okay, let's move on to the next section. And the next section is talking about what actually happens going across the plasma membrane. So the transport of material, to and from, right, across that plasma membrane. So we're going to see that there's different ways that we can do this. Passively, right, which means quietly. Actively, right, which is going to require some energy. And we're going to talk about some of the ways that this happens. So let's go ahead and get started. So let's start with our, first of all, right, the movement of this substance is going to movement of any substance across the plasma membrane is going to happen in one of two ways. Either passively, right, passive means quietly, right, so it's going to require no ATP. Remember, ATP is energy. Right, so passive means no energy, and active transport requires energy. Okay, so there's your difference. So let's start with the passive. The first type of passive transport, there's several different kinds that we're going to see. The first part is diffusion. Okay, so basically what we're going to see is we're going to have a movement of molecules from an area of high concentration to an area of low concentration. So we're going to go from high concentration, the brackets mean concentration, to low concentration. Excuse me. All right. We call this the concentration gradient, okay, the way that it moves. And we're going to see that it happens in everything, gases and liquids. Solids obviously don't move, but gases and liquids, and it's going to keep this constant motion going. Okay, and this happens in both living and non-living animals. So if we look at diffusion, so here we've got, I love this example, so here we've got an eyedropper full of blue dye and a beaker full of water. So we put one drop in there. What happens, right, you see this drop sinks to the bottom, and over time, right, you see it's really concentrated right here. The water around it, right, doesn't have any blue dye, so it's of low concentration. So what starts to happen is that dye basically starts to bleed out into the water, right? So you see it's going from an area of high concentration and it's filling into the water, and over time, right, the whole thing turns blue. So all of those little atoms, right, of the blue color are now just diffused, right, across that medium. So there's three kinds of diffusion, of course, that we can see. We can have simple diffusion, okay, which is just the movement across. So simple is, well, simple. You can have channel-mediated diffusion, okay. So channel-mediated, right, so here's your channel, right, and the ions you see are going through. This is simple diffusion right over here is A. It's just moving across that phosphobilipid channel, right? That's what we see here is moving through. It needs to go through that hallway. And sometimes some of these cells that are trying to get, or these particles that are trying to get into, the molecules trying to get into the cell, need an extra step essentially of security. And we call this carrier-mediated diffusion. So this needs to have a carrier protein with it, right? This is part of that lock and key mechanism, right? This thing's going to come in here. If it fits the right shape, then it's going to be able to go in. So three kinds of different diffusions that we see there. Going from easy over here on the left, easy, to a little bit more complicated. Then we can also talk about osmosis. This is another type of passive transportation. Osmosis is, again, passive, so it requires no ATP, right, at all. We don't need any energy. And what we're going to see here is water is moving from a high water concentration to a low concentration, okay? So this is really best explained by the picture, so let's take a look. So if we start here in our beaker, first of all, we have a membrane here, selectively permeable, meaning it's going to allow water across. Molecules aren't going to really go across, but the water is. So we see we've got a concentration of the molecules on side B, and side A is nothing but water. what osmosis will do is, right, B is really concentrated, right? There's a lot of material in there. So in order to break that up, the water is going to move across the membrane into that area of high concentration to help lower the concentration. So what's important about this is you can see that it changes the volume, right, of this region. So this can have really important implications for your cells because it can easily change the volume of your cells. Then we can talk about tonicity. This is the ability of a solution to affect a cell's tone or shape by altering its water content. Basically what we see here, right? We're altering that water content, so we need to describe what's going on here. So we can have three types of solution. We can have a hypotonic solution. Hypo means low. So here we have a low concentration of material and a lot of water. Hyper means we have a lot of, right? So if you're hyper, you have a lot of energy, right? This, we have a high concentration of solutes, a little bit of water. Isotonic, everything's equal. We have the same amount of solutes and water. So let's take a look at a picture, because again, I really think pictures do everything the best. So if we look at hypotonic, right, you see this cell is really big, right? There's a lot of water inside that cell. Very few, right, particles that you can see are sitting in here. Lots of water. Eventually what will happen is it'll be like a water balloon where if you keep pumping it full of water, it's going to explode. Hypertonic, right, if we take a look over here, hypertonic, you see this is a cell that's dehydrated, right? There's not a lot of water in there. The cell is physically shrinking in size, so it's more concentrated with the material that's in there. And then isotonic in the middle, you have the right amount of water going in and out as solutes. So the cell is going to remain unchanged. It's going to keep its original nice shape, and everything is in balance. All right, let's pause there, and then we'll go take a look at the active transport mechanisms here in a second.